second part of chapter 5, and we're going to start going through some of the different organelles that you find in mostly in eukaryotic cells, although when I say that, we're actually going to start with the plasma membrane, which is technically not an organelle, but is a very important part of the cell. So actually every cell, prokaryotic or eukaryotic, is surrounded by a plasma membrane. So of course it's, holding the cell together, but is also very important in terms of how things get in or out of a cell. Everything's got to pass through the plasma membrane. Attachment to other cells would begin at the plasma membrane, sending and receiving signals. So at its heart, the membrane is a phospholipid bilayer, which we've talked about in a previous chapter. Right? So remember, you have these phospholipid molecules that have hydrophilic head domains and then hydrophobic fatty acid tails. And so they will, in an aqueous environment, spontaneously form this bilayer that is the backbone of that plasma membrane. And there are other components, proteins and things like cholesterol, but we'll do a little bit of a deeper dive into plasma membranes in the next chapter. But yeah, just keep in mind phospholipid bilayer is the basis for that membrane, and also wanted to get across the idea that the membrane is really very dynamic and fluid. So for example, these phospholipids can constantly change places with each other, right? So it's almost like the surface of a lake or something like that, where you could imagine, something could float around within that plasma membrane. So I may have mentioned a few of these already, but importance of the plasma membrane, it's that selectively permeable membrane barrier, allowing certain things in, certain things out, important for maintaining homeostasis, for communication, for adhesion. So lots of critical roles that the plasma membrane plays. Another organelle that you find in basically every type of cell would be ribosomes. I already showed you the cartoon of the prokaryotic cell and pointed out the ribosomes. But ribosomes are very small organelles. They are not membrane bound, meaning they're not made of membrane. But as I mentioned before, these are organelles that produce proteins. And so what are the ribosomes made of? Well, they're really two main components. So there's RNA that's called ribosomal RNA, and then there's a bunch of different proteins. So we bring together these up to 50 different proteins and this ribosomal RNA and assemble it into the functional ribosome. And we're going to talk about protein synthesis in some later chapters, so we'll look a little bit more closely at the structure of a ribosome there. But for right now, just be aware they're small, they're not membrane bound, and they're made of this combination of ribosomal RNA and protein. And you can also see in the image down here, there's really two populations of ribosomes in a cell. Sorry. There are what are called free ribosomes, which are floating around in the cytoplasm, and then there are ribosomes that are part of what's called the rough endoplasmic reticulum, or RER, it's abbreviated here. But we'll come back to this when we talk about the endoplasmic reticulum in a minute. But all this blue stuff in the image here is the ER, the endoplasmic reticulum, and you can see some of it is covered with little red spots. So those red spots are the ribosomes. Okay, so we've got the plasma membrane on the outside of the cell, we've got ribosomes, and then in eukaryotic cells we of course have a nucleus, which is typically the largest organelle. Sometimes in plant cells the vacuole will actually take up the most real estate. But we all know the nucleus because it contains the DNA, right? So basically anything that's happening with the DNA, like DNA replication, the process of transcription, making RNA copy of our genes, that's all happening in the nucleus. I just mentioned that when we were talking about ribosomes, that ribosomes are made out of this RNA called ribosomal RNA. Well, that RNA is produced in such large quantities inside the nucleus that there's actually this sort of densely staining part of the nucleus called the nucleolus that you can see on when you take pictures with an electron microscope. That is where all this ribosomal RNA is being manufactured, and the ribosomes are beginning to be assembled here in inside the nucleus. So we've got our DNA, which is usually in the form of what's called chromatin, and that term just means that the DNA is associated with proteins, actually bound to proteins inside the nucleus. The nucleus is a membrane-bound organelle. It's surrounded by what's called the nuclear envelope, which is just the fancy way for saying the membrane that makes up the nucleus is the nuclear envelope. It's actually a double layer of membrane, and it's interrupted by what are called nuclear pores. So those are literally pores through the nuclear envelope, allowing things to get in and out of the nucleus. And you may also notice here from this cartoon the ER membrane, which is what's in orange here, is actually continuous with the nuclear envelope. So the ER membrane basically flows right into the nuclear envelope. So they are physically attached to each other. Here's a little close-up of one of those nuclear pores. You don't really have to memorize the structure, but you can see there's a number of proteins that come together to make up this pore. And these can be constricted or open, so the nucleus can have a little bit of control over what's getting in and out of the nucleus. You'll also notice here there's some little filaments on the interior surface of the nuclear envelope. We'll talk about those here in just a second. It's basically just kind of helping maintain the structure of the nucleus. So I mentioned that term chromatin, right, which is the fact that, referring to the fact that DNA, your chromosomes are associated with proteins in most instances. So, when we learn about DNA, we learn about that sort of, quote unquote naked DNA, right? Just the double helix, two strands of DNA. But in the biological context, usually that naked DNA has then been wrapped around various proteins and condensed and packaged. So, when we think about chromosomes, it's usually in this format, which is where that DNA has been very densely condensed just to make it easier to move around. A couple of other nucleus-related terms. The nucleoplasm is basically what's surrounding the DNA, the chromatin, and you can think of the nucleoplasm as just this sort of scaffolding network of structural proteins that's really just kind of helping keep the DNA organized. There's also what's called the nuclear lamina, which I already pointed out in a previous image. That's that sort of structural protein that's just on the interior surface of the nuclear envelope, helping the nucleus maintain its shape and preventing it from collapsing in on itself. Okay, so while we're talking about the nucleus, I want to bring up this idea of what's called the endomembrane system. And so this is a system of membrane-bound organelles found in eukaryotic cells that are all either connected to each other physically, so physically touching each other, like the nucleus and the endoplasmic reticulum, as I just pointed out in the previous image, or they're connected to each other through what's called vesicle trafficking. So vesicles are little membrane-bound packages that could move from one membrane-bound organelle to another. So when I brought up the plasma membrane, I was trying to get across the idea that it was quite fluid and dynamic. So, the phospholipids are constantly moving around and changing places with each other. And because of that fluidity, you can have membrane actually bud off of one source, form one of these vesicles, and then that vesicle could fuse with another membrane. So it looks something like this. So here's kind of the overview of the endomembrane system. You've got the nucleus up here in the corner, right, with the nuclear envelope. which is physically touching the endoplasmic reticulum, which is what you see here in orange. You can have some of these little vesicles. So here's a vesicle, here's a vesicle, here's a vesicle. You could have membrane budding off of the ER, forming these vesicles, and then those could travel to, for example, the Golgi apparatus. And those vesicles, when they arrive at the Golgi, can actually fuse with the Golgi membrane and become part of the Golgi apparatus. So even though the ER and the Golgi are not physically touching each other, they are connected by the movement of these vesicles. And anything that happens to be contained within those vesicles could get moved from ER to Golgi. And then as you can see here, you've got vesicles budding off the Golgi as well, and in this case moving to the plasma membrane. Those membrane-bound vesicles can fuse with the plasma membrane, and as you can see here, deposit their contents outside the plasma membrane, outside the cell. So all these components, nucleus, ER, Golgi, plasma membrane, another component that's not shown here called the lysosome, would all be considered part of the endomembrane system and are all connected either directly or through movement of vesicles. So we already talked about the nucleus. Let's talk about the endoplasmic reticulum here for a minute. And if I go back to the cartoon, you can see the ER is literally just folds of membrane, right? That's essentially all there is to it. Either wide, flat folds or sort of skinnier, more tubular folds over here. But it is made of membrane, has a very large surface area because of all that folding. And as I pointed out on an earlier slide, some of the ER is covered with ribosomes, and so we call that the rough ER, right? Because it looks physically bumpy and rough because of all those ribosomes on the surface. And of course, because there are ribosomes there, what do ribosomes do? They produce proteins, right? The rough ER is a major site of protein synthesis. And then, so here you can see the rough ER, right? All those ribosomes. And then in contrast to that, you have what's called the smooth ER, which is smooth because it doesn't contain any ribosomes. And so in terms of proteins being made on the rough ER, the reason for that is the proteins that get made on these ribosomes would actually be produced and sort of injected into the ER. So those proteins that are made here are going to end up inside the ER, kind of trapped inside that membrane. And that's in contrast to proteins that are made on the free ribosomes, right, floating out in the cytoplasm. Those proteins would then be free, right, floating around wherever they happen to be made. And so in the smooth ER, obviously since there's no ribosomes, we're not making any protein there, so that begs the question, okay, well what is the function of the smooth ER? And there's a few important things going on there. So there's some key metabolism, metabolism of certain drugs, for example, happens in the ER. And you could actually tell, looking at the cellular level, you could tell if somebody, for example, was an opioid addict because they would have more smooth ER. Your cells would actually start producing more smooth ER to try to help your body metabolize the drug. Breaking down of glycogen, so remember that's that storage sugar, being able to start release some of that energy and get glucose in your bloodstream. That would happen in animal cells in the smooth ER. And then because the smooth ER is a membrane, is a lipid, it is a source for certain lipids and steroid molecules, right, which remember are also considered lipids. So got production of lipids, bottom line, at the smooth ER. Then we have the Golgi apparatus. Of course, it's got arguably the coolest name of any of the organelles. If you look at the cartoon here, the Golgi is also made out of just like flattened folds of membrane, right? Almost kind of looks like a miniature version of the ER, essentially. Let me go back. But you can think of the Golgi apparatus as sort of the post office, the like shipping receiving center of the cell. So packages will arrive typically from the ER in the form of these vesicles, right? So the vesicle package arrives at the shipping or the receiving dock of the Golgi, which is called the cis region. Basically, just this side of the Golgi is for receiving. So these vesicles are received, they fuse with the Golgi membrane, and now whatever was inside that vesicle that came from the ER is now inside the Golgi membrane. It will typically get modified in some way there, so maybe we add or remove like a sugar or something, or a phosphate group, whatever the case might be, modify the contents in some way, and then we have the shipping portion of the Golgi over here called the transface or the trans region that's sending out new packages or vesicles. And a lot of those might be destined for the cell surface, the plasma membrane, but they could go other places in the cell as well. Excuse me. The last component of the endomembrane system I wanted to mention here are what are called lysosomes. These are organelles that don't tend to get talked about as much as some of the bigger names like the Golgi or the ER, but lysosomes are essentially digestive organelles. And if you look at the cartoon here, here's a lysosome. It almost looks just like a vesicle, and really it is. It's just a vesicle that contains special digestive enzymes. So if there is maybe, for example, food materials that are being taken in from outside the cell into a new vesicle, we could fuse that vesicle containing the food particles with the lysosome, and now those digestive enzymes would be able to start breaking down the food particles. Or maybe there is a part of the cell itself that is damaged, maybe an old mitochondria or something that is not functioning properly. Lysosomes could also be used in that capacity. This phenomenon, what's called autophagy, self-eating. So you could basically fuse lysosomes with that defective mitochondria, and it would be broken down and digested. So that's not uncommon. All right, I'm going to stop there, and we will pick this up in part 3.